Yes. So it's an honor and privilege to be introducing today Itai Yanai, who is coming to visit us from New York. It's not your first time in Tampa?
It is my first time in Tampa.
Okay.
It is.
Okay, so you didn't come for the AACR meeting in March?
I was in the AACR, but it was in New Orleans.
No, the one on cancer evolution.
I didn't go to that one.
Okay.
No, I should from now on. Yeah.
First time in the collaboratorium?
Yes. First time?
In the collaboratorium?
This is my first time in the collaboratorium. It's amazing.
It's an impressive.
You should applaud.
So very briefly, Itai is a professor at NYU, where you had a systems biology lab, right?
Yeah.
And you have a PhD in bioinformatics and a bachelor's in computer engineering, which is up to here relatively standard for field. But also a BA in philosophy of science. So that's a.
Thank you for not laughing.
A lot of people here have a PhD, which stands for philosophy doctorate, but philosophy is not something we do, right? So you are actually a philosophy doctorate who can talk about philosophy.
Not a doctor, but like undergrad.
Well, you are a PhD, so you're a philosophy doctor.
Oh, I have a PhD and I also did philosophy.
Yeah.
I see where you're going. Yeah.
It's very complicated.
Right.
So yes, you have a lot of appointments and extensions. You developed your career in the United States. You did a postdoc in Technion, right?
No,
in Weizmann Institute.
Yeah.
But you were assistant professor at Technion in Israel, and then you decided that New York was cool enough for you.
They offered, and it would have been a shame to turn them down.
Lots of prizes and awards. The prize for academic excellence in the Technion, fellow at the Radcliffe Institute for Advanced Studies in Harvard. You seem to be impressed with your CV. I am impressed with your CV. Many grants, many R1s and U1s. You're a member of a number of study sections as well. I don't know how you find the time to do all these things. On top of that, you have your own podcast.
Night Science, yeah.
That's right.
I'm talking about that.
Yes. So I don't know if those thoughts about night science are going to percolate your talk today.
Not directly, but if anybody wants to talk about it, we can.
Oh, excellent. You have only a few hours in Tampa before you have to rush back to New York, but
Have to talk about it at night.
yes. Anyway, we have to finish soon because I know Heiko wants to watch the German match.
It's two, right?
Yeah.
Thank you so much. Thank you. So it's really a pleasure to be here. I was so happy to get an invitation here because for me, this place is like the mecca of cancer evolution worldwide. I think what you guys have built here is really amazing. It's very progressive. I think the rest of the world will come to realize much later what's going on right now, but you're at the right place, and thank you for hosting me today. So what I want to tell you about is the notion of cell states. Now, when folks here do mathematical models, we tend to talk about resistant and sensitive cells, states. But what I want to interest you in today is to peer a little bit deeper into that. Like, what if you take the hood of the car, let's say the car in this analogy is the cell, and like you just open and you look inside the hood, are there more states than we imagine? Now, cell states are crucial in biology. I don't have to tell you, just think about development, right? When you were a single individual cell, you then differentiated many cells, and the cell states was manifested in terms of the structure, the function, and the plasticity, right? Because there were different cells, so there's different structures. Your muscle cells are different from your skin cells. They each carry out different functions, and there's a certain plasticity involved because a particular cell can then transition from one state to another. So cell states and the plasticity among them, those are crucial concepts. Now, 7 years ago, my lab and I, we started studying cancer for many years studying development. And in cancer, you can think about cancer as kind of like a shadow world of development. And there are many commonalities because, again, there are cell states, there are different states, there are different functions and different plasticities. And there are great commonalities between development and cancer, but there's one big difference, right? What's the big difference between cancer and development? Is that in cancer, the cells harness the greatest force in the living world, which is natural selection, right? And I don't have to tell you, the real reason why across the street people succumb to, across the bridge, I should say, succumb to cancer is because there's evolution happening. That's the real reason that people die, is because there's an evolutionary process. And so that's a pretty big difference between cancer and development. You could argue that there's also evolutionary processes happening in development. There's the adaptive immune system. There's a drive in the germline. You could also like speculate about other instances, maybe yet to be discovered. But cancer is really different, where it's the driving force underlying the adaptiveness is natural selection. So what I want to tell you today are two stories. Tumorigenesis and drug resistance, where the natural selection could be happening not at the genetic level, but the non-genetic level. There's no question with anyone reasonable that you want to talk to that evolution is happening at the genetic level. Like just the observation of I say anyone reasonable because you can always find some crazies, but it's not controversial at all. There's natural selection at the genetic level. What I want to tell you about today, I wanted to provoke you into thinking that natural selection is also happening at the non-genetic level, at the level of cellular states. That's what I want to tell you. So I'll tell you about two stories. No. Oh, again. Oh no, it's because I have to say got it.
Oh, see.
So the two stories. Okay, good. The two stories I want to tell you about are what happens in the normal physiology of the tumor. There are many, many cells, and I'll tell you about how there are different cell states. And then part of this has already been published under this title, but another part has not. It's kind of like the continuation of the project that Mayan Po is leading in the lab, and it has game theory aspects involved, and kind of like agent-based modeling, and it's unpublished, and it's kind of like a crazy idea, and I really want to get your opinion about it. The second is now, how do the cell states change upon treatment with a drug? And this is on bioRxiv. It's not published yet. Actually, it's since the bioRxiv drop, we've been collaborating with Andrew and Alicia, and we're. I say it right? Yeah. And sorry. Very, very nervous. And so it's exciting now to talk about this with you guys. Okay, so we'll get started. Good. So I'll tell you how I came into cancer. It was, I mentioned development, but it was also another side, which was gene expression. For the last 20 years, I've been studying gene expression. It started with DNA microarrays that I designed and loved, and now it then became RNA-seq. But then we were studying the nematode C. elegans. We were studying the embryo, and there was a particular cell that was making a cell fate decision, and we were going crazy trying to figure out what exactly that cell is expressing. And Surani in England had published a single-cell RNA-seq method in 2008, but it wasn't working for us. And we were going crazy because we had spent so many thousands of dollars binding the enzyme. But through that frustration, actually, we developed our own single-cell RNA-seq method. It was published in 2012, and it was the summer when two other methods were also published, START and SMART-seq. And those three methods really did wonders for the field. It started the whole single-cell RNA-seq revolution. It also did wonders for my career, to be honest. You asked how I got the job at NYU. So it was great. And it allowed us to now answer our question of what is happening with this individual cell in the C. elegans embryo. And then we realized, oh my God, it could do so many more things. What can single-cell RNA-seq do? Well, when we looked at the pancreas, you could just take pancreas, mush it up into a single cell suspension. Now look at a dimensional reduction of the cells. And then what you see here, I would call the first law of single cell RNA-seq, which is clusters equal cell types. It didn't have to be that way. That looking at how specific genes are expressed, this is just transcription, it's not proteins. But for some reason, biology decided that transcription is a great marker for states. And when you do dimensional reduction, you see that these are the delta cells, the delta cells, these are the alpha cells. Okay. When you look at tissues where there is also a developmental process unfolding, I would say that you now can describe the second law of single cell RNA-seq, which is that variation within a particular cluster, within a particular cell type, reveals different cell states. So here, this is spermatogenesis study where we took the male germline, again, dissociated the cells into individual single cell suspension, processed it using single cell RNA-seq. And okay, you see this cluster, so this is like, again, the first law, this is a cell type, right? But what about this? This cluster has structure to it. It's not just a circle, it has a structure to it. What does it mean? Well, these are all different states that are stages along the differentiation process of a spermatocyte into sperm. And it led to the notion of pseudo-time, where even though we collected this from one sample, there's no time, it's just one thing, right? You can infer time. You can see that, oh, these cells, they're about to become the same state as these cells are right now. Okay, so single cell RNA-seq was so powerful, and we said, oh my God, we can now study different things like cancer. So when we applied it to cancer, here's just one kind of anecdote that I think will really give you a good description of what's happening. You take a tumor. This is from a collaboration with Rich White in the zebrafish. This is a melanoma tumor from a fish. Take the tumor, dissociate into individual cells, process it for single cell RNA-seq. You see, again, the first law, the different clusters are the different cell types. Here you have the fibroblasts, you have keratinocytes, okay? One notable big cluster is the cancer cells. Now, when we were studying this, a lot of people were focusing on just the immune system. They were trying to look at what states are the T cells in like a hot tumor versus a cold tumor. And it was kind of thrown out to study the cancer cells. The cancers were supposed to be, okay, they're just the same, and they were garbage. And one person's garbage is another person's treasure. And that's what it became for us. We took them and subclustered them, right? So you could just take them, now do a different dimensional reduction study on them. And you see that it's not an amorphous cloud. There's structure, right? There's like something's happening here. There's like meaning. So you could say, well, why are some cells here and some cells there? What does it mean? It could be different things, but what it turned out to be is that these cells, these cancer cells, are expressing different genes than these cells. In a sense, it had to be that because this is based on gene expression. But what if you look deeper in, it's a set of genes that are exclusively expressed in one. So the reason why these cells are here is because they turn on a slew of genes. And here you can look at them. So here, every column is a cell. Okay, like we just take these cells, this is the column. Every row is a gene. And you can see that these cells over here, these cells are these cells. And what's happening with them? Well, they're expressing these genes. You see all these genes like Jun, FOS, UBB. They're expressing genes that are simply not on in the other cells. Okay, so what did we say? There's a population of cancer cells, and there are subpopulations where each subpopulation is turning on a different gene module. Interesting. This could be just an anecdote. It could be unique to weird melanoma tumors in zebrafish, or it could be general, right? Turns out it's general. And this is a review that we wrote where we looked at all the different single-cell RNA-seq papers that studied tumors and specifically analyzed the cancer cells. Like they didn't throw them out like people used to do. And what they see are states. In every single study, people have found states. So, okay, so by now it's accepted that whenever you have a tumor, if you take the cancer cells, it's not a homogeneous population, but rather there are states. Interesting. Okay, so then what we wondered was, is there a kind of logic to it? Is there a rule regarding which states there are? And are some of the states sort of general in the sense that any working tumor has to achieve these states? Is there rules and principles? So what we did, this is in a study that was published over the summer. We took samples that were isolated in our medical center. We're at the NYU Medical Center. And we weren't very picky. We just said, give us whatever you got. So if gynecological tumors were available today, like breast cancer, ovarian, endometrial, we said, come, we'll study them. If we have a colon adenocarcinoma, PDAC, kidney or prostate cancer, whatever, just give us everything. We wanted to do a pan-cancer analysis. And then for every cancer sample, we analyze it separately. So here, these are just the cancer cells from, we call it NYU ovarian cancer 1. Okay, so just one patient's cancer cell. Every column here is a cell, and we're trying to find the modules that underpin the states. And let me pause for a second and say something like a little philosophical. It's not really philosophical, and like the way that you like it. It's that our operational definition of what is a state. You could get like philosophical, like what is a state? Does the cell know what's in the state? We have a very simple operational definition. It's that a particular gene module is on. And so it makes things simple for us. We kind of invert the matrix. Instead of trying to cluster cells, we're clustering genes. And so going back to this, what we wanted to do is find clusters of genes. So for example, here's a cluster of genes. These genes, like all these genes, about like 50 genes, they're on in these cells, but they're not, the same genes are not on in these cells. Interesting, there's like, heterogeneity. So we call this a module. You can see that we found this module, 2, 3, 4, 5, 6 modules from this patient, okay? Now, we do the same thing for every patient. 6 from here, 5 from there maybe. It doesn't have to be the same modules, but 7. We have to take all these modules and then we see, are they similar? And so here's how, this is kind of overwhelming, but we'll just walk through it. Here, the units of analysis are not individual genes, but the modules. Basically, we want to say, hey, this module, the set of genes that we found in this patient, is it the same kind of module, the same set of genes that we found in another patient? So we just say, what's the overlap? Is it more overlap than you would expect by chance alone? You could use the hypergeometric distribution, just to say, is it more? And red means that it's way more overlap. Like these are like essentially the same sets. There's no way this is by accident, like if you just randomly pick a set of genes. So here you see a bunch of modules that came from different patients. It's colored by the cancer type. So you see, regardless of color, the same kind of module was picked up, right? Like maybe one module from patient 5, another module from patient 7. And so we call this like a meta module. And then we can look at which genes are here. And by looking at the genes, you can kind of guess what's going on, because it's not the first time people have seen these genes. You can look at the literature and you can say, oh, this is just cycling. You can annotate the module. And that doesn't mean we really know what's going on, although here it's pretty obvious that if you have a population of cancer cells, some are cycling, some are not. That should leave a signature on the transcriptome. So it's pretty obvious. With other ones, you guys can take issue with why did you call it this? And I'll agree. We don't know. We just look at the genes and we kind of like infer. So cycling is a kind of a positive control. It wouldn't be much of a tumor, right, if the cells aren't cycling. Other ones, I don't know. Remember that module we saw in the previous slide that was called stress? We wrote a whole paper just on that one because we showed that it's not an artifact. You could worry that the reason why you see a stress signature is the way you treat the cells, dissociating and making a single cell suspension. But we show even using spatial transcriptomics and other methods of hybridization where the sample gets frozen right away. So there's no time for a stress signature to come up. It's real and it's universal. Every time we take a tumor sample, there's a stress-like state. There's interferon response, hypoxia, oxidative phosphorylation, metal, complete EMT and partial EMT. So we call these biological process. And again, but what's kind of interesting about it is that, no, not again, because I'm saying this for the first time now. What's interesting is that we're taking tumors from very different cancer types, different patients, obviously, and we're seeing the same modules come up, same states. That's interesting. More interesting are these, I think. These are 5 ways to be epithelial. And most of the cancer types we took are epithelial-based cancers. So there's alveolar, basal, squamous, glandular, ciliated. These are all programs that epithelial cells kind of have access to. And so we identify them, and they transcend the cancer type. So even if a cancer type is originally in the glandular state, like in the pancreas, the cancer cells will then turn on the alveolar and the squamous state that they have access to. And some are brain specific. We also put in brain samples. Okay, so basically there's this catalog of states that recur across cancers. Okay, so everything I've said, if you kind of spaced out a little bit, just to recap, the only thing I've said is this: there is heterogeneity in the cancer cells in terms of which genes they turn on. And that heterogeneity is not random because it recurs across different cancer types and patients. Here's what it looks like for one patient. So this is a dimensional reduction.
Can I ask a question?
Oh yeah, absolutely.
So just so it's clear in my head, you made it clear that the clusters show cell types.
Yeah.
And then you're also saying that within those clusters, there's subclusters that show cell states.
Yeah, exactly.
But you can draw a nice line for those distinct clusters, but the line for cell states is less clear. Is that right?
That's absolutely right. Absolutely.
And so what I find interesting is the size of some of these clusters is very different from others.
It's true.
So
it's true. Some are more universal than others. Right. Yeah.
So I was going to say, do you think that talks to their universality? So if
Yeah.
you've got a bigger cluster of genes that's uniformly
It's more universal.
Yeah.
That's a really good question. So I think you said 2 interesting things. One is that cell states are harder to delineate. And if you think about the variation that we're seeing among the cancer cells, it's not cell type level differences, it's cell state differences. The cancer cells, it could have been that when we do a dimensional, let's go way back here, that when we take just the cancer cells, it would look like this. It could have been that each cancer cell relative to another cancer cell is as different transcriptomically as 2 cell types are. We didn't see that. We see it more like this. It's more like a difference in grade than of type. Second interesting thing you said is that some modules are more universal than others. And that's true. I wonder why that is. I think any tumor we see has the stress, but not any tumor we see has the metal. Metal it's still like more non-random than you would expect. It's still like recurring. It's just not universal. Maybe it's because we just took a snapshot of time, right? Maybe over time, maybe it's more temporal specific. Maybe it's a particular interaction. Maybe it is, maybe only the gynecological cancers have a particular state. It could be something like that.
or maybe it's context.
the context of the microenvironment, like only if you're close to neutrophils,
You're in hypoxia, you're,
only if you're in hypoxia, do you have hypoxia?
How many patients were there here?
You're only 27.
And what kinds of,
Well,
what
actually,
types?
it was 27 that we isolated ourselves, and then we added from the literature 23
Okay.
more.
And then, okay.
Oh, yeah, sure.
And then also, if you go forward.
Yep.
I don't understand the colors, like I can see on the y-axis, the colors, but the top. I see that, but what is going on I don't understand.
What are these different colors?
Yes.
Oh, yeah, sorry. These colors are not by patient, they're by cancer type. So it's these colors. So all the gynecological are one color. Yeah, it's
Okay.
clearer in the paper.
So that, and then the bottom, the red, what are those?
This, good, I didn't say that. This is overlap to the consensus module, to the meta module.
Okay.
Because we delineate this, but then you could ask, how similar is each one to that? Very good question.
Is this all the transcriptome or a subset of genes that's
Subsets, very small subset. It's only about 1,500 genes altogether.
and then like I just want to kind of clarify the definition of cell state, because obviously if you look at lung epithelia versus breast epithelia, they are predictably distinct, and so would you define this still as distinct states? Because it depends on how you slice it. There would be different gene modules, even though they would nicely fit into these clusters, but then they will be quite different if you look at all other modules expressed in these cells, where this. So
in lung cancer, you could take different
well,
modules.
not even like, let's think about it, even normal tissue, like lung tissue or lung epithelia versus breast epithelia versus prostate epithelia. These are not identical.
Right.
Even if you look at sort of basal versus luminal,
Right.
luminal cells from the breast are different from luminal cells in the lung. And so the question is like, would you still define them as sort of substates? And what's basically is the definition of the state? Where does it end? And
So yes, your question is, we're really mixing very different things here. How is it possible to find like a core module?
what more of a kind of what exactly is a state? So here you define the states in this sort of based on these modules, but if you take like different modules, I mean, if you just do hierarchical clustering of or T-SNE of like lung epithelia versus breast, they will form different clusters, even though you might still kind of define these states within each of them.
Right.
So how would you think about these two? I mean, you wouldn't call it different cell types, well, they're epithelial type, luminal epithelial in both cases, but what would you still think of these as different states, or how you categorize this?
I think a very useful trick for us was, and I glossed over it. I hope did you guys catch my trick? The trick was to do a separate analysis for each tumor, and then do the comparison across these very different. Your point is well taken. These are really different. Like one is lung, one is very different. So if I were just to put all the data together, then it would cluster by patients, then it would cluster by is this ovarian, is this endometrial, very different. So here we were proxying for what we wanted to identify was the similarities. We wanted the similarity. So what we did this is a useful trick. I think it's good for all of us to know this trick. It's just move to a different level of abstraction. Instead of putting everything and comparing at the level of gene expression, work separately on each one, get to the level of just lists of genes. This is just a list of genes at the end, and then do the comparison at the level of lists of genes. It's a good trick. Now, if there's something that's very unique to the lung, well, we're not looking for that right now. Because we're only proxying for what's recurrent.
I mean, I understand. My question was more about like about the definition to make sure that sort of.
Okay. So what is our definition of cell state? Let me show you it this way. And I don't think we have a good definition of cell state. I'll tell you what we do. Basically, for every cell, like this cell right here, we score it for each of the modules. Okay, so now we have like 15 scores. Okay, how much is it scored for basal? Not very much. Squamous, yes. Partially T, yes. Cycling, no. Okay. And the cell then is kind of schizophrenic in the sense that it could be, it may seem like it's in multiple states, right? So for us, again, the operational definition is which modules are on. And then it's up to you. You could say, well, is it in this state or is it this? And it's kind of in both. But I do want to point something out to you here. Look at these cancer cells from one patient. Half of them are basal, the other half are squamous. Partially in T is kind of peppered onto it. Some are cycling. So you see that there's overlapping, but some are also mutually exclusive. It's interesting.
Are these binary or are they like a continuous probability that you're assigning?
Yeah. So here it's hard to see. It's some are kind of like in the middle. It's not just on or off. But
Okay. So it's like a probability of being basal or not.
no, it's just it's just a score.
Yeah.
Just a score.
Yeah.
Okay. Now, what does it mean? What does it mean? Like, okay, so we've said that we can use this nice technology called single-cell RNA-seq to delineate gene modules and states. We can map the variability. What does it mean? So the second thing that should have lit up in your mind, the kind of red herring, was that the first thing we do is dissociate the cells. That means you lose all context. Maybe if we just knew the context of which cell is next to which cell, maybe that could give us a clue as to what's happening, what the cell states are doing. So for that, we turn to spatial transcriptomics. And what we do with spatial transcriptomics is we take this is the same tumor for which we have the single cell RNA-seq data. We also have, it's not obviously exactly the same cells. What we do is we take the tumor, cut it in half. One goes for single cell RNA-seq, the other gets embedded in this thing called OCT. You can section it, take that section, put it on a barcoded array where now, you see each of these spots, every spot you get a transcriptome, and it's the size of about 5 to 10 cells. So it's not single cell resolution, unfortunately. But you could think about each one of these as kind of like a nano environment. Like if the whole thing is the micro, then each spot is kind of like the nano environment. And you can characterize it as either malignant, normal, or both malignant, both cancer cells and non-cancer cells. And that we do with just computationally. We look at the transcriptome. We have the single cell data, so we can kind of infer, but it is an inference. Okay, and then when we do this analysis, I'll skip a lot, but basically we get a map where for every state, like the stress-like state, interferon, alveolar, we ask what cells of the tumor microenvironment is it close to? Is it star of the show? The ciliated, like when cancer cells are in the ciliated state, they tend to be close far away from fibroblasts. And the confidence here is because we did this over 10 different tumors, so it's not a lot, but it's also not just one. And for example, cancer cells that are in the interferon response cell state, they tend to be proximate to macrophages in space, also proximate to T cells in space. Interesting. So we wanted to test that. We made mice. We didn't make them. Like we had mice that are RAG1 deficient. So that means they have no T cells. Okay. So now we've perturbed this aspect of the microenvironment, and we want to know. Okay, if you perturb the microenvironment, does it also lead to a perturbation of the state composition? Are the cancer cells now in a different state? And we saw that it does. So I'll walk you through it. So here. We just repeated everything in mice. Mice, if you take it's kind of obvious. You probably are not surprised. If you make tumors, just like we saw in the zebrafish, like we saw in human, there are also cell states. They're kind of overlapping. And look at this. Here, if you look at the fraction of cells that are cycling, it's about the same whether it's in a wild-type mouse or in a RAG1 deficient mice, right? But the interferon response cell state is much less frequent in those mice that don't have T cells and B cells. Interesting. You perturb the microenvironment, you perturb the states. Kind of makes sense. So, what did I tell you so far? I just want to conclude kind of like what was in the paper to tell you the new idea that I really want your feedback on. The idea was that there are states that you can best score them using the notion of gene modules, right? These gene modules are not novel. The cancers didn't invent them per se. They seem to be co-opted from existing modules. This is like the shadow world concept of, if you look at them, it's stuff that development uses. It's physiological, it's a biological response, like hypoxia. It turns on HIF1 alpha. Like we already had HIF1 alpha. Cancer cell just induces it. So it's all co-opting modules, and likely these modules are not genetically encoded. Likely. I haven't shown it to you, but I think it's some evidence that we have to consider that you take many different patients and they all have kind of the same states. It's probably not genetic. It's probably genetic. And that leads to this notion that there might be a kind of epigenetic, I shouldn't say that word, there might be a kind of non-genetic takeover, where a lot of the oncogenes are happening early on, p53, all the good stuff, right? And then you got the tumor, and then from that point on, it's not genetic, but non-genetic variants that can now be under selection. So that's an interesting idea. I'll build up on it. So just to summarize, modules are best way to make states, that they're co-opting developmental context. It could be like with the microenvironment that's involved, and not genetic. Yeah, question?
Yeah, I actually had the question that you just said, but I will just ask your insight a little bit more on that. Like you said, you didn't find anything specific to cancer, like new modules. But you have a way to find the novel cancer unique modules with your, or did you work on that and you really couldn't find anything new? Because now the thing is, you take your gene signature and compare it with the literature, which is biased already coming from the normal cell modules, right?
Sure.
It will always connect to the things and you will maybe miss those new modules already. Do you have any,
Yeah, so the notion is, I think you're hypothesizing a cancer-specific module, maybe even recurrent, like all cancers sort of converge to this cancer-specific module. We didn't see something like that, but maybe we did and we just didn't know it. I'll give you an example. Hypoxia. Why do we call it hypoxia? That we have a list of genes. We call it hypoxia because there's HIF1 alpha in there and like the usual suspects, right? So we call it hypoxia, and we think we understand. We say like, oh, good job, team. We got this one, nailed it. But we also observe that when we grow cells in a dish, they have plenty of oxygen. They will also turn on the hypoxia module in response to a drug treatment. So maybe they're not using the hypoxia module in the way that we think they're using it. Maybe it is cancer specific, but they're just using existing genes. I mean, it kind of like, it would be crazy for the cancer cells not to use existing pathways, right? Why would it not? Why would it keep away from using the things that are there? But it could be tweaking it and
going, exactly, or maybe due to the origin of cancer is sort of genetic disease, right? Some genes are all either activated or inactivated. So the pathway, in fact, has to change if it is used in this particular cancer type. And actually it is observed, I think, for example, IDH mutant glioblastomas, they use strict acid cycle in a different way, sort of, just because IDH is mutant. Now the metabolite going into the strict acid cycle there has to exit, and now the exiting metabolite has to be utilized in a different way.
Right.
So for example, I think
that makes perfect sense. Like you take an existing module, then tweak it a little bit. That's what I would do if I was the cancer.
So somewhat related, but I think it's like very, very important. Like if you do a thought experiment and you were to include normal tissues from the same kind of organs, I would assume that they would sort of cluster fairly close to cancer ones in these modules, but would you expect to see anything unique or some clear and sort of repeatable distinctions in the
Great question. Great question. In other words, to what degree are these modules unique to the cancer, or would you just take any normal, like if we have just done this, not on cancer, same exact analysis, but on normal tissue, would we get the same? Great.
Yeah, I mean, there's evidence actually that there's like in breast cancer, like basal cancer cells cluster much closer to normal basal cells compared to luminal cancer cells.
Primary, and then the metastasis.
Yeah, the metastasis is kind of.
All bets are off.
But this would enable perhaps to see if there's any kind of recurrent themes that discriminate cancer.
So what we showed in the paper is that yes, in normal non-cancer populations, you do see many of these modules. It's just that it's at much lower levels and not at the same level of heterogeneity. So for example, if you take the pancreas, then all of the epithelial cells will be in the glandular epithelial state, almost nothing in squamous. But in PDAC, now half of them have become squamous. So that's the, but now you take like a lung sample where there are cells in the squamous state, they'll all be squamous, and only in lung tumors do you now see the different epithelial states. So these are all found all the time in normal. It's just in cancer, it's kind of like more, higher scoring and more heterogeneity. Good question.
Is more cell-to-cell heterogeneity or is more promiscuity of different?
More cell-to-cell heterogeneity or more internal to the same?
That is more promiscuous
Good
in
question.
terms of gene modules.
Yeah, it's both. Even within each cell, the average number of modules that are scored, if they're cancer cells, is higher than scoring normal cells. Yeah, thank you. You guys are awesome. You guys are good. Okay, so
Can I just make one more comment? Because I think you said something really important about the mutation opens the door for plasticity. Is I think what you said. Is that what you said?
that the mutations open the door to plasticity. I mean, you can get plasticity without mutations too.
Yeah. But I think you're implying that in cancer, that you need these key mutations to start.
Early on. It's kind of like the Christina Curtis Big Bang model. The mutations. Yeah, I mean, we all know it's going to be crazy for anyone to argue, like we said at the beginning, that it's not genetic. The genetic evolution is not involved, right? We see the P53. We see all the usual suspects. The brilliant point that Christina Curtis made is that it's early on. Mm-hmm. Now, what happens later? That's really interesting. Could it be that non-genetic evolution is where natural selection is acting?
But is that what you're saying, or that's what you're wondering?
I think both. I think it's. So what this is based on, the piece of evidence is that we're seeing the same modules recur across different patients. That to me is evidence against the genetic level. It could, but I haven't shown it formally. Okay, so.
Because you brought up this term multiple times, what is genetic evolution for you?
Genetic evolution is that if I sequence the genome, I'll see a mutation. What's it for you? It's good to agree on. It's to agree on terms.
Because I would argue selection does not act on the gene. Selection doesn't know that there's a P53 mutation.
Of course. But just over time, we see the frequency of the allele go up. Just in like a more.
I would also agree with that, but I would ask you, why is that? It's not because of the mutation. It's not because of a base change.
Well, I take a very, as you'll see in a minute, a very kind of Dawkinsian point of view, selfish gene, where it's really a bunch of alleles, and the cells that carry that allele will have a different fecundity, different chance of survival, and then over time will feed back to the gene. So that selection is occurring at the level of the gene. But then I think we will get a little too soon. Yeah?
Sorry.
That's okay. Don't apologize.
So based on your previous slide, when you are actually, clustering everything from different patients, this kind of implies some sort of a universality of the states over time, right?
Right, that it's stable over time, yeah.
Yes, but the question is, if you are, would it be possible, because, cancer is growing, some specific genes are going to be selected, some states are going to be selected. Would it be possible to take some patients that have been, say, having tumors maybe earlier in disease, later in disease, and cluster them all together to see the transition?
Yeah, and then you could ask, what is the first state? How do they, is it maybe the first cell has many states and then they kind of, sub-functionalize?
Yeah.
Or, we're doing an experiment now with PDAC and mice. We can't do it in the same mouse because, once you sack it, it can't. But we're hoping they're fairly similar from individual to individual, and then we're sacking at different time points. So we'll be able to see. But yeah, it's really interesting to see the dynamics. And then also metastases, as we kind of hinted, is like a whole different game. And it's interesting to know whether maybe the heterogeneity is restricted to the primary. Maybe in the metastasis, it's really just one state. Now it's just like EMT, right? Because it's all EMT. Okay, we're good?
Yeah.
Okay, so what would it look like then if the evolution is happening not just at the level of the genes, but also at the level of non-genetic aspects like these states? So what do we know what must happen for us to invoke natural selection? Of course, it's that there are variants, that the variation is heritable, and that there's a correlation between the success of the variant and having the variant. So if we can somehow show that states, we have a population of cancer cells and they're in different states, if we can show these three aspects, then natural selection must follow. That would be kind of cool, right? Well, first of all, we know this because that's what I just told you. We know that there's variation. Okay, so that's an easy one. Heritability, we can do experiments where we ask a mother cell, what's her state? What about the daughter cells? Are they in the same state? What about the granddaughters? How stable is it? We can do experiments like that. I won't belabor the point, but we do this by barcoding. Right, so every cell has a kind of lineage barcode that when we sequence the transcriptome, we also recover which barcode it has, which is just stable. And then we see heritability. We see that all the cells with this barcode, they're in the same transcriptional state. And we also did it with organoids. This is still ongoing, but with organoids, it's really cool because do you notice that you can passage the organoids? It's kind of like a sponge. I'm originally kind of like a developmental biologist, and what's cool about a sponge is, it's an animal, and you can put it through a sieve and now get single cell suspension. Right? Leave it, and it will reconstruct the sponge. Same thing with the organoids. You can passage them, and then again they will form organoids. But now every cell is now in a totally different kind of context, right? So you can ask, despite this dramatic thing, are they still in the same state? And we see some evidence that they are. So there is some evidence. It's not still ongoing. I don't want to tell you that I'm concluding it, but there's some evidence that it's heritable.
Can I ask a quick question?
Yeah, of course.
So for some of the states, the heritability makes sense.
Yeah.
For others, it's a bit more difficult.
Yeah, you're absolutely
Your
right.
stress or hypoxia, right? If you're in hypoxia, you're unlikely to be in a cycling state. But to get offspring, you must change into a cycling state.
Cycling, right.
So hypoxia, for example.
Thank you so much for saying that. Thank you, because it's something I should have said. So thank you. I'm very grateful you said that, because what I want to tell you is, and I kind of suggested it before, but now I want to really highlight it. There's two classes of states we feel. This is our working model, is that once one class is from here to here, we call this biological process. It's kind of like normal cell physiology. It's just cycling, hypoxia. Like there's no oxygen, it's going to go into hypoxia. Or maybe it's doing something different, we don't know. So this is like probably not heritable. Probably not. But then there's this other class that we're really excited about. We call the second class cell identity. These, we have some evidence showing, require chromatin rearrangements. It's not so simple for a cell to plastically transition into another cell. Like it's kind of like more dramatic to go from glandular to squamous. We have some evidence that like chromatin rearrangements have to happen. That flavor, that class of states we feel are more likely to be heritable. So when I say states from now on, I want you to kind of think about these. Okay, these we expect. Okay, so now some evidence that it's heritable, but what does it do? What do they do? How much time do I have? I feel like
Another half an hour.
it's 12:57. We started at 12.
Yeah, but
We've done a lot of progress.
there's going to be questions.
Oh, so we can go until 1:30.
Some people might leave at one,
Some people
but
leave.
yeah.
Feel free to leave, although the best is really It's not my fault if you missed this. Okay, so no, I didn't mean like that. I didn't mean like that. So okay. Look, I want to present a kind of idea. I ran it by Andrew and he gave me some feedback. I'm very grateful. It's kind of a wacky idea. A lot of assumptions are going to be happening, but just indulge me a little bit, okay? Just a little thought experiment. So when there's a cancer cell and it's interacting with a cell from the microenvironment, maybe think about this as a fibroblast. So now there's a cancer cell and it's close to a fibroblast. Like let's imagine this interaction. Like I like to think like I'm the cancer cell, what would I do? I'm coming into contact with the fibroblast. I would say that there's a fundamental asymmetry between this interaction. It's not a fair interaction. Why? Because whatever the fibroblast does, it's programmed to do. It's just physiological response. Cancer cell. Okay, initially it will also have a sort of physiological response, but the cancer cell really has the upper hand. The cancer cell, because it's part of a population, can undergo evolution, natural selection. So it can evolve a new response. In other words, in this interaction over time, this side can evolve, this side cannot, therefore it's fundamentally asymmetric. Go along with me on this. And already I'm sure there's a lot of, whoa, easy now. But if this is the case, you can think about the cancer cell manipulating this cell in a particular way. Now, how would it do that? And as I mentioned earlier, I'm a Dawkinsian. I'm a fan of Richard Dawkins. He wrote The Selfish Gene, which is actually what got me into biology from computer engineering and philosophy. I read that book. And then he has another book called The Extended Phenotype. And it's on David's shelf, right? Yes. We saw it. The Extended Phenotype, he says this. An animal's behavior tends to maximize the survival of the genes for that behavior, whether or not those genes happen to be in the body of the particular animal performing. So what does it mean? He gives a few examples. Here's a nice one. Imagine a snail, and the snail now gets parasitized by a worm. Okay, so now it's got a parasite, and it's been observed that when snails get this parasite, they have a thicker shell. Their shell becomes thicker. Interesting. Now, the snail is doing a kind of economics before it's getting infected. It thought about how thick the shell should be. Too thick, it's investing a lot of its resources into just defense, resources that could be spent on reproduction, finding a mate and all that. So that thickness is like a trade-off, right? It's well thought out. Now, the parasite comes in. The parasite doesn't care about the snail's reproduction. The parasite just wants to be protected. So it kind of makes sense for the parasite to want the snail to have a thicker shell. It will secrete some factors that will lead to a thicker shell. Okay, so now, given the situation, we all agree that there is a gene for the snail having a thicker shell, right? There's a gene for having a thicker shell. What's interesting is that that gene is not in the snail, but it's in the parasite. So the thicker shell is an extended phenotype of the parasite. Interesting, right?
But what do you mean that? Basically, it means that there must be a mechanism to regulate thickness of the shell that the parasite can manipulate.
Yeah.
So it's not that the internal,
it's not-
external.
right. The snail already had all the machinery, right? But in the same sense, you could say, let's say I'm redheaded, okay? So what is it? It's I can get a mutation. I don't know exactly how it works. I should know. But I think it's a single mutation, isn't it? Shouldn't say mutation. Say allele.
Don't go into red here, it's actually much more complicated.
Much more complicated. Let's say what's like a single change. Well, but the notion with hair, what I wanted to say is there's the whole machinery for how to make how to make hair. This just the gene. Maybe it's interesting that you say it's more complicated because to make it red, it could just be that you're using the same machine that's existing, you're just modulating it a little bit. In the same sense that the parasite is just modulating a little bit.
Yeah.
And so we use that genetically all the time. Here, it's just interesting that the allele is not present in the organism that bears the phenotype. So can this be applied to cancer? Well, it's not novel at all to say that the cancer cells manipulate the non-cancer cells, right? That's well known. For example, there are mutations in cancer cells that can lead fibroblasts to then make the environment immunosuppressive, right? The cancer cells can like effectively say to the fibroblasts, hey, please call the T cells off, right? So we know that cancer cells shape their microenvironment, but here's the sad complaint that I have. Very sad complaint. I want to show you that this argument though doesn't make sense for the level of cell states. On the face of it, it doesn't make sense. Why? Because let's say the one particular cell state. So this is the whole population of cancer cells, and some subpopulations in this particular state. And it's now creating a public good. But the benefit will go back to all the cells, not preferentially to the state that creates it. If that's the case, it can't work. That doesn't fulfill the third requirement for natural selection. The third requirement is that the owners of that particular allele should get preferential benefit. If they don't, it's not that sad. It's just that we can't meet the third criteria. And then we can't say that there's natural selection happening at the level of cell states.
Well, unless you consider space.
Yes, exactly. So what we want is a model where there is a preferential benefit. And Andrew's point is exactly right. You did it a lot faster than we did it in the lab. But okay, here's the punchline of all this. It's going to go into a few details. It's going to be fun. Well, I'll just say the punchline and then the question. So the punchline is going to be that it does make sense if you remember that cells are physical entities with neighbors, and if you add the constraint of neighborhoods, there is preferential benefit. Okay, question, and then I'll show you the details.
Now I'm processing your last sentence.
Oh no, I'm sorry.
I'm still stuck a bit on your cancer fibroblast. I mean, you're trying to force a linear thinking on a non-linear problem, right? Because, your cancer cells have only evolved the ability to mutate or to change the fibroblast behavior because fibroblasts were there.
Right.
If you put these cancer cells in an environment where there aren't any fibroblasts, I don't think your cancer cells will evolve a trait to manipulate fibroblasts.
Right.
So sure, the fibroblasts have change the cancers. It's a chicken and egg thing, right? I just don't think is it a cause or is it causality or correlation? It's
So
a
I
bit
think,
difficult.
okay, let's say you're the fibroblast, I'm the cancer cell. I come to you.
I'm the fibro?
You're the good guy. You're the good guy.
Okay.
You're the good guy. Okay. And now, because of my presence, you will change, right? You will. And because of your presence, to me, I will change. So we both have like a kind of like knee-jerk effect, right? Right. But there's more. Things are going to unfold over time, right? It's not just for one day. We're going to be living for years together, right? Because we're living over.
So you evolve a trait to manipulate me.
But you cannot evolve a trait to
That's
manipulate me.
fine.
But that's all I'm saying.
But if I were not in the room, you would not learn the trait to manipulate me. So my presence shapes your evolution.
Yes, I agree with that. I agree with that. Oh, I agree with that.
So when you're looking at a microenvironment, let's say the liver, you know, you have cancer there, and you have all your microenvironment, say fibroblasts.
Yeah.
You have some level of heterogeneity within them. Say it's rather small, but then so if you want to compare that between like a healthy person, the liver microenvironment with fibroblasts, do you know how the heterogeneity changes between no disease and disease?
Yeah.
Because I'm wondering, like, how certain are you that the fibroblast is not this population is not evolving?
How certain am I that the fibroblast population is not evolving? Well, first of all, you're absolutely right that the composition will change in the face of cancer. Like we've seen that there are more neutrophils. There's less endothelial cells. Like there are like the cancer cells do kind of like warp the reality.
Right.
But how confident am I that the fibroblasts
Or individual
themselves are not
cell
evolving?
types? Individual cell types.
Well,
Yeah, from a normal to disease state of the patient. Is there a change in heterogeneity?
I think that the fibroblasts, you know, one argument that's intuitive to me is that the fibroblasts are dividing very slowly. So just that prevents them from evolving. If though, you will now show me a fibroblast population that has variants, that has heritability, that's going to be easy, right? If it's genetic, and a correlation to survival, then natural selection should act on them. But then you'll have to kind of agree with me that they're pre-cancer, right? They are becoming cancer then. Yep. And the fact that we don't see that suggests that really only one side is evolving. Okay, so let me show you a simple simulation. This is a game of life like situation. We drew inspiration from Game of Life, where black. This is a board, and black means no cells. And then there's two kinds of cancer cells. There's the orange and purple, and this. There's also two kinds of microenvironment. There's pink and white, and if you just evolve it over time, then nothing kind of happens. Or sometimes here the orange will win, but it's just luck. If you play it again, the purple will win. So nothing's happening. But let's hypothesize that so there's two cancer cell states, the orange and the purple, right? Let's hypothesize that whenever orange is close to a fibroblast, the squares are like the microenvironment, then a benefit will come. And that benefit will come to everyone. So you see this fibroblast, it's been activated because it got the signal. Let's say it's paracrine signaling. It got a signal. The benefit goes to everyone. So it goes to, you could say, well, it's not preferential because benefit goes everywhere. But does anybody want to say why is it preferential after all?
What's the good?
What?
Why is the good as distributed? They distribute closer to certain cells, right? It has diffusion rate, it has a decay rate, so it's going to be more present next to the cells.
Right.
There's more orange around.
There's by definition, an orange
Yeah.
next to the activated one, otherwise it wouldn't be activated, right? By definition, there's at least one, but can there be zero purple? There can be, right? So as this is playing out over here, look at, this is the simulation now.
Unless it's well mixed, probably not.
Well,
No, we see that generally, so it will, the orange now, the one that has a kind of extended phenotype relationship, will always win now. So it does work. So theoretically, at least, it does work. Now, what's interesting is that you can now hypothesize that there's multiple extended phenotypes. In this case, the orange can activate, but also the purple can activate another element. Like maybe these are macrophages that get activated with the purple. And what's interesting there is, do you know who wins? If this has an extended phenotype association with this element of the microenvironment, and this one has an extended phenotype association with this one, who will win? No one wins. They become balanced. And this one was, I guess it should be expected, but I didn't think about it, that perhaps the reason why we see recurring states is because every cell state achieves a kind of neighborhood, a microenvironment for it, and then together they can secure their own survival. So this is, we're testing this now, we're doing co-culture experiments in all kinds of ways. But I think what's exciting about it is that we see these states, we're not sure why are they recurring, what is their stability, but because there's evidence for their heritability, and because there's evidence for the way that they can get preferential benefit, we think that might explain their evolution. Yeah.
That is only because the distance and the intensity of those effects are the same, right? The orange that when it affects the fibroblast and the other one when it affects the
Yeah, but it's only like a cell.
that is only because there's the same.
What if the parameter was that this arrow extends much farther? Right. That's true. That's true. It is a feature of this model, but it's true. I would just say that paracrine signaling tends to be very ubiquitous here. So that the signaling is not of the kind that we see in development. Is also an morphogen signaling. You know, like who's in Drosophila beacoid?
I was thinking of Turing, but
What?
Turing. I was thinking.
Yeah, Turing effect. Right. You could hypothesize that. Here we're hypothesizing the inverse. But you're absolutely right. It's an assumption. It could be that it's not true. Absolutely true.
I have a question. So, why is investing in having a macrophage and fibroblast and Tregs, if all of them have to be competing? Is that the reason they are there? Is that because everyone just support a niche for the neighborhood that you just said?
Right. I think so. I mean, see, the alternative explanation that gets promoted is that I think if you look at like the Axelrod paper of cooperation, the classic paper, I think that that one is not so spatial in its thinking. The underlying assumption is that they're cooperating. How are they cooperating? They're each coming up with a good, and now I create some good, you create some good together. We can kind of like divide and conquer. Instead of each of us having the hallmarks, we divide the hallmarks. So that's one model. The thing is, I don't think that makes evolutionary sense. Like theoretically, it doesn't seem to make sense because each state needs to get a preferential benefit to secure its own survival. So what I like about this model, I think it's plausible, is because theoretically it makes sense. Okay, I think
I was just going to ask, out of all your cell states, how do you know which ones are extended phenotypes and which are just normal phenotypes?
That's a really good question. I think some states don't seem to be heritable, so they filter themselves out.
Yeah.
And the only ones that could be candidates for this are the ones that have some stability, which are the cell identity ones.
I think there is evidence that even cell cycle state can be heritable actually, and especially for cells that have stayed out of cycle for a prolonged period of time, when they eventually redivide, the daughter cells can also be less proliferative.
makes sense. Yeah, that makes sense. You're right. Well, so some cells could just be kind of like it's heritable that they're more they're in a proliferative state, and that's transgenerational. Okay. Yeah, you're right. You're right.
If you think about the
So when you, on your previous slide, when you first introduced space, and this, no, the next one, I'm sorry,
This one?
where you showed preferential benefit to the orange clone, right? And if you were to introduce a cost of manipulating your environment, right? Because now your orange clone invests energy in manipulating the environment, which it doesn't invest into proliferating.
Incur a cost.
So if you infer a non-zero cost, will the orange still win?
Depends on the benefit.
Depends on the but still.
Cost must be less than the benefit.
That's what I mean. If the
But
cost
I love
is
that you
higher
say
than
this because it gets me into like a game theory. I think this needs to be more like properly game theory.
because then the likelihood of having more purpose cells adjacent to the provider of the good is higher.
Yeah.
Or the other way around, the orange is lower.
Yeah. I'm just happy because it's obvious you guys get it. That's good.
Interesting to do the analysis of when this trade-off happens.
So I think there's like one perhaps elephant in the room, because like if you think about the evolution, it's competition between sort of different kind of heritable lineages, clonal lineages, and I mean all these things there would be kind of correct if the states are kind of true types. And the thing is that they can be seen by selection, because selection acts on phenotype, but there's also this phenomenon of cells of state switching. So if you have a cells in the similar phenotypic state but sort of different lineages, they're really kind of competing with each other, and you don't care necessarily about the survival of the same state from a different clone. On the other hand, if you have this state switching, you might be able to kind of get kind of best of multiple worlds depending on where you end up to. So I think there's this additional layer of complexity if you incorporate the possibility of state switch and then context dependence of the fitness.
Yeah, it's really interesting. I mean, at first I was really afraid of the idea of state switching because in my mind it's okay, now it just generates a new variant. But then I thought two things comforted me. One is you could say that a state is quasi-heritable and then compute to what degree does the model still hold. Like maybe it's like 80%, 70%, maybe even 60% heritability, it still works. So it could be a quasi, maybe we don't have to insist on full heritability. But second, maybe the whole state switching itself is a variant, right? Like maybe that is a strategy and we can model that as the unit. That's actually more interesting.
Yes, exactly.
That's what you guys would think? Oh, yeah.
Just on that, but I want to ask another question. We've looked at the idea of memory in cell states. And so that could actually answer that question, right? There's a period of time where it'll persist, but then it can reset. And so that could switch to something else. And that could be tied directly to protein levels.
Yeah.
Right. But the other question I wanted to ask was, since you have spatial transcriptomics, you could actually go and see where your cell states are.
Right.
And is there patterns in or clusters, spatial clusters of those cell states, and have you done it?
Yeah. Right. So it's ongoing right now, but we do see evidence for neighborhoods. If you say, okay, these states, ciliated versus alveolar, is it salt and pepper, like randomly, or is it more neighborhood?
But you've shown this data earlier. Right?
Yeah, and we do see neighborhoods. I think what would make me have more confidence in it is if I could toggle it, if I could go in and I'll like make a change and see that now. So
and that was my question was it's powerful, but what states do you see pre and post treatment? And if you're using the pancreatic mouse model that you were talking about, the tumors are so consistent between the control and the treated groups, you could actually ask, what states are go away upon gemcitabine treatment of that animal versus the control. like which are the most important for resistance or
Right.
sustainability during treatment as well.
Yeah. So there is a whole part of the talk that more and more doubting that we will get to. But yeah, there are new states that emerge, and also the elimination of other states. It's just that most of our work has been in vitro. We do have an in vivo component, but it hasn't been. It's kind of like a different project in the lab that doesn't talk to this first project in the lab. So I have to integrate them better.
The fact that you identify neighborhoods is not direct proof that you have a cancer cell modifying the cell that it interacts with, because even if you look in normal settings, like, hematopoietic stem cells reside preferentially near the osteoblastic niche because there they have the right nutrients and they're protected, and that's not evidence that they are actually doing something to the osteoblast. It's just evidence that they find that environment comforting.
Yeah, it's like a correlation, not causation. But what for you would be? Because I'm trying to think. Like seriously, what is the killer experiment to show? The ideal would be to figure out what is the mode of signaling. Like if it's paracrine signaling, okay, then what exactly is the soluble?
What if you put normal and cancerous cells near the same support cell type, and you could look at whether the fibroblast or whatever cell type that is closer to cancer cells than to normal cells in the same well or whatever is actually changing its gene expression more than the fibroblasts that are closer to normal cells are.
Right, but to do that in a dish?
Yeah, why not?
Well, because in a dish, what's a normal cell in a dish? Because they're all like immortalized, like a cell line.
Normal cells, like more normal cells for sure that grow in dishes, right?
Yeah, the best would be somehow in vivo, but then it's like.
But in vivo, you cannot control the proximity to the cells, I guess, because
No,
you cannot control the
wrestle with this all
ratios of
time.
the two
We
different
keep trying to
types.
fight. What is the right experiment? Do you want me to quickly run through? Oh, you have a question?
Oh, I was just gonna say it might be even more complicated than that, because you could have coexistence within a neighborhood and still have the same mechanism of preferential fitness. Like the orange doesn't have to win out in a public goods game, right? You could still have coexistence even though there's still preferential fitness, like what Heiko is saying.
Right.
If there's a cost.
Right.
Yeah.
We all agree it's complicated, I think. I think we all agree.
Please proceed.
Please proceed, okay. I wanna, we have, I think, 8 or so minutes?
Sure.
7.
We'll give you 10. How about
Another minute. All right. No, I'm just kidding. Okay, here we go. So I'm gonna just try to quickly tell you a story. See how fast we can do this. So this is about a drug treatment, and we got inspiration from something that was done with bacteria and antibiotics. Basically, Roy Kishoni, when he was at Harvard, made this mega plate where he grew bacteria, but in one region there's no antibiotics, and then here there's kind of like a small concentration of antibiotic, but still enough for them to not be able to get there, the bacteria. Here 10 times the concentration, 100 times, 1,000 times. Right? You've seen this before? Raise your hand if you've seen this. Oh, so you know. Okay. So basically, you see them evolve before your eyes. And it does give us evidence that the dose is really important. It also gives us evidence that the time is really important. So dose and time, we're really inspired by that. But then if you look at models on drug resistance, they don't seem to take dose and time so much into account. The models are either that the resistant state was pre-existing and it got selected for genetically, or that some cells were primed, like non-genetically, to survive. So Gustavo in the lab, he set up this experiment where basically to recapitulate the Kishoni experiment, by making these 10 lines. This is a Kuramochi cell line. It's an ovarian cancer cell line, and it's treated with a PARP inhibitor. Oh, this has been blocking it for you the whole time. Is that?
It's on ours.
Oh, it's on yours. Oh, it doesn't matter. So we have now cells that got the dose of the drug, and it took them 10 days to now achieve confluence. And then we take a subpopulation, we almost double, 2.5. Gustavo did this, and he liked 2.5. And then it took them here 17 days to achieve confluence. He freezes an aliquot of this, so we can always go back to it. Doubles the dose, doubles. This is like a really high dose, 320 micromolar. It's like a ton of a dose. But now we have these lines. And as many people have shown, including Andrew here, because I clicked here.
The time is all over the place though.
Yeah.
What's all
13,
of-
15, and 12 feet and 14.
Yeah, isn't it interesting? Here it took 50 days. We were like, come on.
Yes.
It took
Yes.
Gustavo a whole year to generate these lines. What's interesting is if you now do the cell viability plot, this is the original cell line.
Mhm.
And you can see that the cell lines that adapted the higher dose are now much more resistant. And so really, you could see that these cells can be really pushed forward. You know, there's no limit to what they can achieve. If you just challenge them, they will meet any challenge given enough time. Just like the bacteria. Well, you guys are all smiling in like a weird way. What's happening? I feel like there's an inside joke. Okay, so now
We've seen stuff like this.
you know all about this. But have you seen this? So we're single cell people. So now we look under the hood, as I like to say, and we've got the subpopulations. Okay. So look at T5. These are the cells that adapted to fibroblast, and you see subpopulations of the T10 subpopulations, and you could say, okay, is this subpopulation of cells? Is it the same kind of state as this, or is this the same as that? So what Gustavo did, he made the average of each subpopulation and then compared the average transcriptomes. And what you see is that there are 5 states. This is now states in a different version. These are drug-resistant states. And so this like meta state, it includes populations from the control, the 1, the 2.5, even the 5 is in here. The next state has 2.5, 5 states. So see, this state only arose, it seems, in the 2.5. And then this state over here, it's got the 10, 20, 40. So it arose in the 10. This state has, 10. Already it was born at the 10. This one only has 160 and 320. It arose rather late. You can look at it evolutionarily that at the beginning, the control is only this state. At 1, there's 1 resistant state, and then a new state, state 2, is born. State 2, you only see it at 2.5. We didn't see it here, we didn't see it. You could say we missed it. I would say, come on. So that's like my scientific answer. and then it rose in frequency, because now it's like 50% of the cells that are in the population that have 5. I'm out of time. One minute. 0, some might say, and so forth. Okay, so I don't want to belabor the point. This is actually just figure 1 of the paper.
Can you find single cells in the 320 or in the higher doses that exist in the 0?
No.
Like some type of stem cell?
They're all 100% in state 5. Yeah.
What if you take the drug off?
If you take the drug off,
Of these 320 cells?
if you take the drug off, if you take the drug off, well, there's 2 aspects. You can look at it at the cell survival. They maintain their cell survival, right? Like this thing, they maintain. They don't go back so much. But there's also apparently a genetic aspect to it. So if we sequence them with whole exome sequencing, there is aneuploidies, but we don't know if it's related because this is a PARP inhibitor, so it's DNA damage, right? We see aneuploidies, but we don't know if it's related. We also see a lot of epigenetics. We also see a lot of non-genetic changes. You're not supposed to say epigenetic. Did you guys get the memo? You're not supposed to say epigenetic anymore. Okay,
Yeah,
so
you don't care. I guess my point was, is there ever any return to any of those other states? That's my point.
Yeah, well, here's what I want to do.
You gradually decrease to the.
Yeah. So, Gustavo doesn't want to hear this. Very against the idea.
This is live on YouTube.
It's on YouTube?
Yeah.
Hi, Gustavo. So, he doesn't want to hear about how we're gonna go backwards. Now you tell me it's on YouTube.
It is live over there.
How about that? Okay, so I just want to flash through that we looked at the genes. This is like figure 2 in the paper. We looked at the pathways. We studied this a little bit more in detail with facts and show that we could, within a play, we could isolate subpopulations. We show it's different from persisters. You've heard of persisters, of course. This is different. An adapted state is different. This shows that there is genetics involved, but there's also non-genetic aspects, because if you do a tax-seq, you can see different peaks are opening, and it's not random. There's particular factors that are probably involved. We also did it in vivo, and the states are also occurring in vivo. So overall, what we conclude is that there's a resistance continuum where the cells can be challenged. The reason, the way, I think this is no news to folks here who are champions of adapted therapy, that a high dose may be disadvantageous because it will challenge the cells to now become resistant. And the more high the dose, the more high the survivors will be resistant to it. It's mediated by genetic and non-genetic changes. Gustavo doesn't know that. And what's really interesting is that if you look at the way they become resistant, it's by changing their basic biology. They're turning on, I just flashed it through a minute, but the states have different biologies. Like, these are different functional pathways that are coming on. And we were talking earlier that maybe a new wave of models should incorporate this biology. That's what I wanted to say. Thank you very much. I'll talk to you later. Gustavo did this part, and Dalia, she led the work on the states that are recurring, and Mayan is now pursuing the whole extended phenotype aspect. Thank you, you guys were great. I'm glad we had an hour and a half. I couldn't have done it. I really needed that other half. Thank you. Thank you. Thank you.
Yes, I'll do it.
I think I know in a triangle.
Trying to think in my mind. And so it really comes down to, well, unfortunately, Joel is not
Yeah, I was like, oh, yeah, my neighbor that not making fire. Surviving. Fire.